Scientists, we are on Unit 2, Topics 6, 7, and 9. Today we're talking membrane transport and facilitated diffusion. So up to this point, we have talked about the cellular membrane with all of its components, its integral proteins, its peripheral proteins, the channel proteins, the glycoproteins, the glycolipids, the cholesterol. Now we're going to see all of that in action. So let's go. So we're talking selective permeability, and I'm going to dump some of this on you just like that so you can start taking notes. So selective permeability is this understanding that some substances can move across a membrane more easily than others. Easy passage across the membrane are things like small nonpolar molecules, hydrophobic molecules. These are the things that'll pass real nicely through the hydrophobic tails of the phospholipid bilayer. Some examples of these kind of hydrophobic small nonpolar molecules are things like hydrocarbons. Those go really easily across the membrane, and a bunch of different gases too. Gases like CO2, oxygen, and nitrogen or nitrogenous gas. Those are all really important for cell functioning, so they can just easily pass through the membrane and get inside and do their job. Things that are difficult to pass through the membrane or things that are assisted by one of those integral membrane proteins are hydrophilic molecules, so loves water, polar molecules, large molecules, ions, kind of like the ragtag team of all of the other sort of molecules needed in the cell. Examples of this could be sugars. Remember, sugars are a huge molecule comparatively, and ironically enough, water. Water has to be able to pass through using a technique called osmosis that we're going to get into. So transport across a membrane, there's two main types of it. There's passive transport, which is just things moving across, like you're able to pass through, no problem. And active transport. Active transport requires energy, most of the time assisted by some kind of protein or channel or pump or something. So we're going to take passive transport first, just because it's a little bit easier to explain and understand. So passive transport. Passive transport is the transport of a molecule, and it does not require energy from the cell, or because at this point the molecule is moving with its concentration or electrochemical gradient. What we mean by that, well, we're going to get into what we mean by that. It's involved in the import of materials and the export of waste. So we need CO2 to come into the cell, or we need O2 to come into the cell, and we need to export, the opposite, right? CO2 or O2. Examples of this, there's three of them: diffusion, which is diffusion across a membrane, osmosis, and facilitated diffusion. So let's recall back to Unit 1 when we were talking about concentration gradients and things moving from high to low concentration. In this process of diffusion, and again, this is a passive transport, it doesn't require any energy, it is spontaneous, right? It results from a constant motion of molecules. So molecules are constantly in motion, and as they move more and more, they want to kind of spread out, right? So if there's a lot of them concentrated in a small space, they're going to want to spread out to where there's not that many of them. I think in Unit 1, I used the example of going to a really packed party, and you just want to go off into the kitchen, and then people start trickling in, right? You're going from high to low. We call it moving down the concentration gradient. So I like this example here. This is like a green sugar cube, right? We put it in some hot water. Those molecules inside the sugar cube are going to be moving around a whole bunch, and eventually they'll start to spread out to the point that it's almost a homogeneous substance here, right? It's almost everything is the same. Now it's not a sugar cube in water, now it's sugar water, right? So it's moving down the concentration gradient. Molecules diffuse differently across the membrane, right? There's different rates of diffusion. There's nothing that's needed. It passes directly through. So you can see here, this is the extracellular fluid. So that's outside the cell. And then the cytoplasm exists inside the cell. This is the stuff that all the organelles are floating in. And you can see here, number one, these little molecules here are going straight through. There's no integral protein. There's no nothing. It just goes straight through the membrane. And these ones are going a little bit at a constant rate, right? So you have almost as many in the extracellular space as you do inside the membrane itself, as you do inside the cytoplasm. However, in contrast, you can see these green little guys over here, and these green guys are passing through the membrane, again, without the help of a protein or a channel or anything like that, but they're going a little slower, right? Eventually, they'll get to the point where it's equal on both sides, but again, the rate is a little bit slower. And remember, even with diffusion, the membrane is selectively permeable. So this could have something to do with the shape of the molecule itself. Osmosis! Love osmosis. So we talked about this in Unit 1 when we were doing properties of water, but we're going to hit it home again. So the diffusion of water down its concentration gradient across a selectively permeable membrane is this process of osmosis. And this is sort of counterintuitive. Here we go. You can also think of it as diffusion of water from areas of low solute concentration to areas of high solute concentration. So taking a look at this example down over here, this is a classic example. You have a U-tube there, not YouTube, but YouTube, U-tube. Anyway, there's a selectively permeable membrane that allows water molecules to pass through, but not sugar. And you put some sugar molecules on one side. So you can see there's more sugar molecules over on this side than there is on this side. Eventually, what will happen is this water level goes up as this water passes through. Think of it trying to like dilute this sugar water, right? So even though there is now still more sugar on one side than there is on the other, the water itself has sort of evened out, right? It's the passage of water from low solute to high solute concentration. So if I was to take a measurement of concentration of sugar in this water and concentration of sugar in that water, it should be relatively the same because the water, again, just went and kind of diluted it. So that is the process of osmosis. It's a little different and it feels a little opposite than diffusion. Great. All right, facilitated diffusion. It's the diffusion of molecules through a membrane via these things called transport proteins. Increases rate of diffusion for small ions, for water, for carbohydrates, right? So these things that can pass through the membrane, but it's a little bit easier if we can find a door, right, to get through. Like we can always climb through the window, but I think a door is easier. Two categories of this are channel proteins and carrier proteins. Each transport protein is specific for substances it can facilitate movement for. So it's like a door that's shaped just for you. No one else can get through it. It is specific for that substance. Why is facilitated diffusion passive transport and not active transport? Well, that's because facilitated diffusion is still passive transport because things are going down their concentration gradient. No energy is required. This is just like a cheat code for the cell, right? We can always climb through the window, but it's easier if we have a door, right? That's facilitated diffusion. It facilitates you on your way in. We're going to facilitate. Ooh, welcome. Come through the door. Okay, so let's talk channel proteins. So this is one type of protein that acts in this facilitated diffusion process. It provides a channel for molecules and ions to cross through. So you saw some of these when we were making our models of the membrane, right? This is an integral protein. but it's an integral protein that has a channel through it. That's all. The channel itself is hydrophilic, allows for that passage, and some of them are gated. Some of them we call gated channels, right? Many are gated channels, I should say. This only allows passage through when there's a stimulus. So we are going to be modeling this in a simulation, and you're going to be able to see it firsthand, where when there's a need for it, when the concentration on the outside of the cell, or for that matter on the inside of the cell, gets too high, these gated channels will open, allowing these molecules to pass through and sort of regain that homeostasis. Aquaporins. Aquaporins are just fun to say. Aqua, water, porins are pores, right? So these are specific channels just for water. All right, carrier proteins. Carrier proteins are similar to integral channel proteins, except they change shape to allow things to pass, right? So you can see here when there's a substance on the outside of the cell that needs to go in, one of the molecules will come and interact with this carrier protein, and the carrier protein will change shape, thus shoving these inside, right? It is still passive transport. There's no ATP that's being used. This is a conformational change. It's like a key in a lock, you know? You don't have to have energy or electricity to put your key in your lock. Carrier proteins, they carry. Okay, in terms of diffusion, describe what's most likely to happen to the particles in the image below. We're going to do this ad nauseum in class. I hope that you said something along the lines of moving from high concentration to low concentration. Represent a plasma membrane. Great. You may even ask here, well, is this a large molecule? Is this a hydrophilic molecule? Can it not pass through here? Where's my channel protein? There's a number of things you could have said. I'm sure all of them were somewhat right, and I'm very excited to talk about it. Not required for facility. Okay, we're not doing the practice problems. We're going to do them in class. Skip over them. So skip over pages 34, 35. Let's talk active transport. So active transport is the transport of a molecule that requires energy. It moves against its concentration gradient. So we're shoving more stuff in there than can actually fit, right? And we need energy to be able to do that. These are things called pumps, right? So we're going to see some protein pumps. There are things called co-transports. That's like you grabbing your friend by the arm and being like, you're just coming with me. You're coming with me. You're coming with me. We're going to get through this membrane. You're coming with me. There's exocytosis. That's moving things from inside to out, like waste products that we need to get out of the cell, or endocytosis, that is bringing things into the cell. Cyto, meaning cell. Esis is a process, and exo is out, endo is in. One of these days, I swear, we got to do prefixes and suffixes. So, out of the cell, into the cell. Let's talk about it. Here's the big one. Oh my God, I love this. ATP. God, I hope this isn't the first time we're talking about this. I am so in love with this molecule. It's so cool. All right. It's the energy source that's used by cells. Adenosine triphosphate. So it's got 3 phosphate groups, right? You can see them over here. 1, 2, 3. That's how you know it's an ATP. Essentially what this does is it transfers this terminal phosphate group, terminal meaning the last one, right? Like a bus terminal. It's at the end of the line, to the transport protein. And when it does that, it changes the shape of the transport protein to move that substance across, right? So you can see 1, 2, 3 phosphates here. Essentially what it does is it, boom, breaks that link. This one over here gets transferred to the protein, says, hey, thanks for the ride, and then goes inside. Okay? ATP is going to come up, adenosine triphosphate. It's going to come up so much this year. It's my favorite molecule. I love it. I find it so cool. I'm trying so hard not to freak out about how much I love ATP right now because I could go on like a 20-minute tangent. Okay, also known as a conformational change, right? That's what we talk about when we say it changes shape. It's conforming, right? Conformational change. It's conforming to some kind of shape that we want it to be. Great. So pumps. Pumps help maintain membrane potential. Membrane potential is this unequal concentration of ions across the membrane, resulting in this electrical charge. So the cytoplasm is relatively negative in comparison to the extracellular fluid. So you have the cell here, right? In the that's my nucleus, and the cytoplasm is slightly negatively charged in comparison to the positive charges that are outside. So what the cell does is it stores energy in these electrochemical gradients, right? So things move down the concentration gradient on an ionic level in the same way that they do a concentration level. So if there's a lot of negative charges inside, we'll move them out. Same thing with positive. We'll move them in until some sort of homeostasis is reached. Active transport is a little bit different though, because again, we're moving against those gradients, right? So there's more negative on the outside, more positive on the inside, or vice versa, and we're trying to pump it across. So some examples, we have an electrogenic pump, right? This generates a voltage across membranes. It might be used later as an energy source for cellular processes. The most common type of electrogenic pumps that we'll see is in neurons, right? Because your activity in your central nervous system is essentially just volts of electricity, right? Being passed from one thing to the next. So then that's how we can see and move. Sodium potassium pump, right? Sodium is positive. Potassium's also positive. We need a pump to get it in. Animal cells will regulate their relative concentrations of this by using this sodium potassium pump. We're going to see it visually in just a second, but essentially 3 sodiums get pumped out of the cell and 2 potassiums get pumped into the cell. And what this does is it creates this charge, this +1, this positive charge to the extracellular fluid, and then boom, we can set off that charge and something happens, right? So this is sort of a way that cells get some kind of electricity. These are the electrogenic pumps. It's kind of a wicked cool process. Again, I'm kind of obsessed with it. So let me just dump this on you. Okay. So actually, I think this is one that would be good step by step. Animal cells have a relatively high concentration of potassium inside the cell compared to the outside. So potassium in this is represented by these orange circles, as you can see up here in the legend. This is the cytoplasm inside the cell, extracellular outside the cell. You can see out here. So there's more potassium inside than there is outside. They have, animal cells have relatively low concentrations of sodium, represented here by the red squares. And again, you can see that there's more outside than there is inside. Right over here, you have the actual sodium potassium pump. That's this green guy. You can see he's an integral protein that hangs out inside the phospholipid bilayer. And you have this ATP molecule that kind of hangs out on this side. Okay, so ATP is always involved in this active transport. So in this shape, when it's pointed inside towards the cell, the protein can take in 3 sodium molecules, right? So 3 sodium bind to the protein itself from the inside to the outside. And remember, we're going against the gradient, right? So even though there's not a lot of sodium in here, we're going to pump 2 of them out. ATP phosphorylates. Phosphorylates is just that chopping off of the phosphate group. That's what that means. Adding it to the protein. It phosphorylates the protein, which causes this conformational change. It causes the protein to change shape. The new shape, right? So we can see here. So there's our phosphate group. This phosphate group came from ATP, right? It's now bound to our protein, so it's in a different shape. And when it's in a different shape, it has a different function. This new shape causes the sodium to go away, go outside the cell, and for two potassiums to come in. Yay, potassiums. Remember, we're working against our gradient, so there's more in the cell than there is out of the cell. That's why we need this guy. When potassium binds to the protein, this phosphate group is released. We don't need it anymore. Sorry, I should go back. So when this phosphate group is released, it's released back into the cell. ATP didn't go away. Remember, matter can neither be created nor destroyed. It must be changed from one to the next. So the ATP molecule will just go ahead and receive this phosphate group back again, right? So loss of the phosphate causes the protein to return to its original shape. So here it is again, ready to get 3 sodiums and trade them for 2 potassiums. Ta-da! That's how we pump things against their concentration gradient. So we have a ton of potassium in here now, and because of this, we have a slightly positive charge outside. Examples of pumps. Protein pumps. Proton pumps. Proton pumps are huge when we're going to be talking about cellular respiration and photosynthesis. It's essentially a proton pump is a membrane protein that builds up a proton gradient across the membrane. It's used by everything, plants, fungi, bacteria, and what this does, literally the entire job of a proton pump is to pump hydrogen out of the cell. We pump it out of the cell. That causes a gradient, more on one side than on the other. Co-transport. Okay, this is your friend grabbing your arm and saying, you're coming with me. You're coming with me. We're going to get into this restaurant. You're coming with me. It's the coupling of favorable movements of one substance with the unfavorable movements of another, right? So grab a friend, let's bring them, right? This uses energy stored in these gradients, generated by the pumps that are pumping out the protons, to move substances against their gradient. So again, it's like you're trying to force your way into a packed crowd, right? And one person can go just fine, but like if you hold your friend tight, they can come too, right? Co-transport through that high concentration gradient. Favorable movement is downhill diffusion, right? So things move from high to low concentration. That's sort of like a downhill movement. Unfavorable movement is like we're trying to shove things, like we're trying to push something up a hill, right? It doesn't want to go that way. I don't know why they just threw these 2 random vocab words in there, but yes, unfavorable movement and favorable movement. It's like, what does the molecule want to do? Okay, plants use co-transports for sugars, for amino acids. One of this is the sucrose hydrogen co-transporter. So sucrose can travel into a plant cell against its gradient only if it's coupled with a hydrogen that's diffusing down its gradient. So sucrose can only go up against the gradient from outside to inside the cell, right? Unless there's hydrogens that are moving in the opposite direction. It's the only way. Look at this cute thing. So here's your co-transport. I can't get through there alone. I'll help you, right? And we're able to have this, the hydrogen, co-transport with the sucrose into the cell, right? And it's able to do that because ATP pumps these hydrogens out of the cell. If we pump enough hydrogens out of the cell, there's going to be more up here than there are down here. And as scientists, we know when there's a high concentration on one side and a low concentration on the other, they're going to get through. Sucrose just kind of follows along for the ride. That's your co-transport. Transport of large molecules done through exocytosis and endocytosis. So think of this as like the cell eating and the cell pooping, right? Exocytosis is the secretion of molecules through vesicles that fuse to the plasma membrane. So this is moving things outside the cell. Exo, again, meaning outside, cyto, meaning cell, lysis, meaning process, right? Something that happens and we move it outside of the cell. Essentially, vesicles fuse to the membrane, right? So you'll have the cell, you got the plasma membrane around it, and the transport vesicle will kind of come and fuse to the membrane, and it forms like this weird double membrane bilayer. Once it's fused, the contents just kind of like spew out into the extracellular fluid. This is, it's a lot of waste product, but not just waste products, right? We're going to talk about cell signaling and hormones and that sort of thing. There's chemical signals that are sent out as well, tells the cells what to do, when to grow, when to divide, that kind of thing. Nerve cells releasing neurotransmitters, right? So your nerve cells never really kind of touch each other, right? One nerve ending kind of is around the other nerve ending, but in order to release a neurotransmitter and have a signal passed down from one nerve to the next, to tell your fingers to snap, right? It has to go through this exocytosis process. Endocytosis is the opposite. It's an uptake of molecules from vesicles fused to the plasma membrane. So there's something on the outside, and it's going to come in and hang out on the membrane, and then it's going to fuse, and then all the stuff's going to get dumped inside. Phagocytosis and pinocytosis is two types of endocytosis. This is essentially like eating and drinking, right? Eating and drinking. So one of them brings in like water, one of them brings in molecules and nutrients that the cell needs. Sometimes we have receptor mediated endocytosis. That's like the thing that's being brought inside the cell needs to have a signal to attach to, like they have to high five before doing it. They have to receive the signal first, and then we go through the process of endocytosis. We're going to look at this in pictures and images in just a second. We're going to draw this. This is going to be a whole thing. Okay, so phagocytosis, when a cell engulfs particles to later be digested. So this is, again, like eating, the cell eating, a process in which the cell is eating. So here you go. So you got the particle here. It's being transported into the cell itself. The cell surrounds the particle. Don't worry about this word pseudopodia. Pseudo meaning like not real, like pseudoscience or pseudo medicine, and podia meaning feet, like podiatrist. So that just means like fake feet. And that's these things, right? That's these things that come up around it, around each side. So it surrounds the particle. It packages the particle, so a membrane will form around it. You can see this here. Membrane has been formed around it. And this food vacuole, so this is considered a food vacuole after we, package it up. It fuses with lysosomes. So here you go. Here's a lysosome right there. It's going to fuse with that. The lysosome's going to put its hydrolytic enzymes in there, and it's going to get all digested and yummy, and then it's going to go into the rest of the cell, and everyone's going to be happy. Pinocytosis is slightly different. This is uptake of extracellular fluid. So this is like you're drinking. Pinot, I'm sure you've heard of like wines that are named Pinot, and then cyto, meaning cells, and then process of. It's an uptake of this extracellular fluid containing dissolved molecules. It's not specific, so really it's just the cell kind of forming these little things here. Pseudopodia. They take in dissolved molecules in a protein-coated vesicle. So again, you're going to get this membrane around here. You got a bunch of proteins that are attached to it. those proteins help mediate the transport of the molecules itself. Remember, we don't want this lysosome to come in here and digest this. This is not for digesting. This is dissolved molecules in extracellular fluid. It's the drink. We don't want to mess up the water. So they have these protein-coated vesicles to say, hey, don't lyse me. Receptor-mediated endocytosis is essentially the same thing, except we need to come down and make sure there's like a lock and a key, right? So it's a specific uptake of molecules with the solute binding to the receptors in the plasma membrane. So you got all these receptors on your plasma membrane. These might be glycoproteins. These might be glycolipids. These might just be peripheral proteins, right? There's a number of things that could be these receptor molecules, right? Pseudopodia comes up, so it allows the cell to uptake large quantities of a specific substance. So if we're looking for just calcium, right, we can have it come and bind to these receptor-mediated channels. When the solute binds to the receptor, they'll cluster in a coated vesicle. And again, the coat is there for to protect it, to make sure that it gets to where it's supposed to go and it's not just eaten up by these lysosomes. And then it's taken into the cell and done whatever it needs to do. Practice FRQ. All right, we're going to be doing some FRQ practice later. So don't worry about this or the practice. We'll do that in class. Cool. Thank you so much. I can't wait to see you in class. That was fun. I liked that topic. See you soon.